For years, this stuff felt like pure science fiction. The idea of a tool so precise it could literally edit the building blocks of life. But what was once just a concept in a lab is now out in the real world, performing what can only be described as medical miracles. So today, let's break down what CRISPR is all about. The thing is, this isn't some far-off future tech anymore. Because in 2023, gene editing took this massive leap, right out of the laboratory and into a real, life-saving cure. It fundamentally changed the game for medicine, forever. And that leap? It's got a name. It's the very first therapy of its kind. And believe me, it's signaling the dawn of a whole new era in how we treat disease. Okay, so let's talk about Casgevy. In late 2023, this became a huge deal. Regulators in both the UK and the US gave the green light to the world's first ever approved therapy based on CRISPR. It's a one-and-done treatment for two really awful blood disorders, sickle cell disease and beta thalassemia. And how it works is just, wow. Doctors take a patient's own stem cells, use CRISPR to edit them in a lab and fix the genetic mistake, and then put those corrected cells back into the patient. Those cells then go on to produce healthy red blood cells. So you see, this isn't just about managing a disease. For so many people, this is a flat-out cure. Okay, so we've seen this incredible result. But that brings up the big question, right? What is this stuff? What is CRISPR? Really? How does it all work? So the full name is Clustered Regularly Interspaced Short Palindromic Repeats. Yeah, it's a mouthful, I know. But here's the cool part. The concept is way simpler than the name. It's not something humans invented from scratch. Scientists actually borrowed this trick from bacteria. It's a natural defense system, basically a tiny immune system that bacteria have been using for millions of years to fight off viruses. We just figured out how to repurpose it. So how do these genetic scissors actually work? Well, think of it like a biological search and replace function. First, scientists create something called a guide RNA. It's basically a little molecule that's programmed to find one very specific spot in all of our DNA. Then, they attach that guide to an enzyme called Cas9. This is the scissors part of the system. That guide RNA then leads the Cas9 enzyme through the entire genome until it finds the exact DNA sequence it matches. Once it locks on, boom, the Cas9 cuts the DNA. And this is where the real magic happens. The cell's own repair crew rushes in to fix the break, and scientists can use that moment to either disable a faulty gene or even insert a new correct piece of DNA, effectively rewriting the genetic code. Now you might be thinking, haven't we had gene editing for a while? And you're right, but the reason CRISPR is such a revolution comes down to this comparison. The old ways, things like ZFNs and TALENs, man, they were clunky, super complex, slow, and really expensive. You had to build a whole new custom protein for every single gene you wanted to target. What CRISPR did was make it cheap, fast, and easy. Instead of building a whole new car, you just have to give it new GPS coordinates. It's that simple. It made gene editing accessible to almost any lab in the world. And because it's so much easier to use, the possibilities, well, they just explode. We're really just scratching the surface of how this tool can reshape our world. Let's start with medicine, because the potential here is just mind-blowing. We're talking about engineering a patient's immune cells to become super-powered cancer hunters. Or, get this, what if we could literally just snip the HIV virus right out of a person's cells, offering a real, permanent cure? These aren't just pie-in-the-sky ideas. This research is happening right now, along with work to reverse certain types of blindness and even tackle huge problems like diabetes and heart disease. But hold on, it's not all about medicine. CRISPR is set to completely change agriculture too. Scientists are using it to create crops that grow more food, to make our produce more nutritious, and to engineer plants that can fight off diseases all on their own. That could be a massive deal for feeding a growing planet. And this isn't some far-off future thing either. In Japan, you can already buy the first CRISPR-edited food, a tomato designed to have higher levels of a healthy compound. And this really just drives the point home, right? Look at the range here. From making T cells better cancer fighters, to editing mushrooms so they don't turn brown in your fridge, which could cut down on food waste in a big way. It's even being used to create super-fast diagnostic tests for viruses. It's less like a single tool and more like a Swiss Army knife for all of biology. Okay, but with a tool this powerful, you know we have to talk about the flip side. The risks, the red lines. Because as they say, with great power comes great responsibility. I mean, you have to ask yourself, when you get the power to rewrite the fundamental code that makes us who we are, what are the things that could go wrong? Right, so first things first, the science isn't perfect. Not yet. The biggest worry is something called off-target edits. That's when the molecular scissors accidentally cut the wrong place in the genome, which could cause all sorts of unintended problems.
And even when it cuts the right place, the cell can sometimes be a little sloppy in its repair job, making new mistakes. There are also concerns about bigger structural damage, and even the possibility that our bodies might reject the Cas9 enzyme as a foreign invader.
And that's exactly why organizations like the FDA have put very strict rules in place. They demand tons of testing and data to make sure these therapies are as safe as possible before they ever get near a patient. It's all about managing those risks.
But even beyond the science, there are these huge ethical questions. And the whole debate really exploded in 2018. A scientist in China announced that he had created the world's first gene-edited babies. The reaction from the global scientific community was swift and furious. It was seen as a massive breach of ethics, crossing a line that many thought should never be crossed. It forced everyone to have a very serious conversation about where we draw the line between curing disease and heading down the slippery slope of so-called designer babies.
So you've got this incredible potential on one side and these very serious risks on the other. Where does that leave us? Well, the most important thing to realize is that this isn't about the future anymore. It's here.
I mean, think about the journey. We went from discovering a weird quirk in bacterial DNA to developing an approved medical treatment that cures people. It is safe to say CRISPR has officially left the world of science fiction and landed squarely in reality.
You know, that approval for Casgevy, that wasn't the final page of the book. That was just chapter 1. CRISPR is now a basic tool in the toolkit for scientists everywhere. And while there's still a long way to go, its story is really just getting started.
So it all comes down to this, really. For the very first time in the history of life on this planet, one species, us, can now consciously rewrite its own genetic code. We hold the pen. The power is in our hands. The challenge now for us, for society, is to decide what story we're going to write.